Hi everyone, in this lecture we're going to complete our discussion of Chapter 2. In this portion of Chapter 2 lecture, we're going to talk more about cell-to-cell communication, or how neurons talk to each other. So let's begin. How do neurons talk to each other, or how do they communicate? It's imperative that they do. This allows you to perform any basic behavior or any reflexive behavior. Here we're taking a look at an individual that's touching something hot, for example, an iron. So first let's focus on the type of neurons that are going to perceive this hot stimulus. These are sensory neurons. These are neurons that are going to bring in information from the environment. They could bring in light information from your visual system, also auditory information or sensation from touch. So for example, if you are touching that hot iron, your sensory neurons that are in your skin would detect that. And we say that these neurons are afferent, meaning that they're going to be bringing information in from the environment to the central nervous system or to your brain and spinal cord. So once your sensory neurons bring that information in to your brain and spinal cord, then messages need to go back out from the brain and spinal cord to your limbs, basically so that your body can react to the stimulus. And how this happens is motor neurons. So for example, your sensory neurons are sensing that there is a hot stimulus that you're touching. That information is sent in to your brain and spinal cord. Then information is sent back out, and your motor neurons will allow you to reflexively pull back your hand from the hot stimulus. Now, motor neurons are bringing information again, or a signal, from your brain and spinal cord, your CNS, to your limbs. So the message or the signal is going away from the central nervous system. So we say these neurons are efferent, or moving away from the central nervous system. So basically it's quite a simple process when we look at it in this way: information in and then information back out. And what is very important here as well are what are called interneurons. Interneurons are located entirely in your central nervous system. And what they do is allow connection between your sensory neurons and your interneurons. So your sensory neurons are bringing information in to your central nervous system, and then information is coming back out through interneurons to your motor neurons to allow your body to react or to move. Now let's take a look at a reflexive response more closely. If we take a look at our individual here at the bottom right of our slide, we can kind of see a very simple version of how this individual would sense this hot object and how that would then allow him to pull his arm back in reaction to that hot object. So first we have the hot object, which is a hot iron. You can see here is his arm, he's touching the hot iron. Now, sensory neurons in his skin on his finger are going to detect this painful hot stimulus. The dendrites of those cells are going to detect that signal of pain or hot. And remember from the first portion of lecture from this chapter, dendrites are what receives messages from either other neurons or from environmental stimuli, like a hot iron. So the dendrites of sensory neurons here on the skin are going to detect this painful stimulus. They are then going to send a signal through their axon all the way to their terminal buttons, and then that signal will be released into the synaptic space, or just that gap between the axon of this cell, or these cells, these neurons that have detected this painful stimulus, and all of the neighboring neurons around it. Those neurons will be able to detect that signal from their dendrites, and then it's a chain reaction. So all of these stimuli are going to react. All of these neurons are going to have this reaction of receiving this signal with their dendrites, bringing that information into their cell body, and then their axon will allow the signal to travel down and be released through their terminal buttons, and then on and on and on until more cells or more neurons are reacting. And eventually, these signals will reach an interneuron, which will become excited or have an excitatory response. Again, that will bring the signal or allow the signal from these sensory neurons to make it all the way into the motor neurons. The motor neurons will then send a signal back to the muscle of the arm, same way, through a receiving signal with their dendrites, bringing that signal into the cell body, and then sending a signal down the axon to be released by the terminal buttons. And these motor neurons sending the signal will then connect with the muscle of the arm, and then the muscle will contract and the arm will pull away from the hot stimulus. So thus far, this whole chain reaction of sensory neurons sending that signal to interneurons, which then send the signal to the motor neurons, which connect with the muscle that allow the muscle to contract and pull back, we think about this as being excitatory. So something is actively happening. Now, there is an inhibitory component to this particular response as well. If you think about it, if you touch a hot iron, you're going to just pull your hand back. But let's think about this scenario really quickly. Let's say you have made a cup of coffee and it's gotten cold and you want to reheat it. You go to reheat it in the microwave and you go to pull it out and it's scalding. It's absolutely scalding hot. You way overdid it, but you don't drop the coffee mug. You hang on to it long enough to get it to the countertop where you can then let go without dropping it and shattering it everywhere. Well, what's going on here is that there is that excitatory reaction happening that we just discussed previously. So there is that signal happening to contract the muscle in the arm and drop the hot mug, but there is also an inhibitory signal happening as well. There is an inhibitory signal of don't contract, don't contract, don't contract, just long enough to get that coffee mug to the countertop. So I highlight this just to emphasize that these signals don't always have to be excitatory. They can be inhibitory as well. We've been taking a look at neuron communication so far on a broader scale. Let's look more closely now. Let's take a look at what is going on the cell membrane or the membrane of the neuron when communication is happening. Now, when a neuron is at rest, it's not sending an electrical signal. It's not communicating with another neuron. It's just basically kind of hanging out, doing nothing. We call this resting potential. Basically, this is when the balance between the outside of the cell and the inside of the cell is such that the cell doesn't react or doesn't do anything. So here we're looking closely and we're taking a look at ions in the fluid on the inside of the cell, which is the intracellular fluid, and also the ions on the outside of the cell membrane here, which is the extracellular fluid. And when these ions are in such balance inside and out, then we basically have a resting potential, meaning that the neuron is balanced both inside and out in terms of ions in the fluid, that it's not going to send any signals. It's just at rest. Now, when these fluids become out of balance, that's when the neuron actually becomes active and starts sending signals. Now let's take a look closer at the composition of the intra and extracellular fluids. Now if we look first on the inside of the cell, so this is the inside of the cell membrane. Here is the membrane again. Here is the inside of the cell. We have A- for short, or organic anions. Negative meaning that there is a negative charge. We also have positive ions, potassium. On the outside of the cell, we have sodium and chloride. Now notice again, we have a positive and a negative. Now I want to take the moment here to kind of remind you all back probably many years ago when you were a smaller kid. They probably had you at some point in class take magnets, positive and negative. If you remember, magnets, they have a positive pole and a negative pole, right? And if you try to push two positives or two negatives together, think about what they do. They repel each other. Negatives and positives attract one another. That's going to be very important as we move forward when we take a look at how these fluids outside and inside of a cell kind of become out of balance, and that leads to the cell becoming active. So how do we measure the charge of the negative and the positive ions on the inside and outside of a cell? We use what's called a voltmeter, and we insert a recording electrode like one here. This will allow us to measure the concentration of ions on the inside of a membrane. This is measured by measuring millivolts. So a membrane potential at rest is around negative 70 millivolts. So what that should tell you is that when a membrane or a neuron is at rest, inside the membrane, that fluid is negatively charged. Now, when this fluid on the inside changes from negative 70 and becomes, for example, more positive because of the exchange of ions in and out of the cell membrane, that will produce what we call an action potential. Now we can take a look at action potential more closely. So what we were just taking a look at, again, we're looking at the concentration of ions on the inside of the membrane. And again, that's measured by millivolts. Here we're looking at the membrane potential. So what millivolts? Resting potential is at negative 70 millivolts. So the neuron's not doing anything. The fluid on the inside is negatively charged, as it should be when it's resting. It's moving along just fine. Nothing's happening. And then the fluid around the membrane on the inside and outside, the ions there start to exchange and come in and out of the cell membrane. And when more positively charged ions come into the cell, it becomes more positively charged. This is called depolarization. So this is going to, again, reduce the concentration of negative ions from negative 70 inside the cell more towards 0. When this happens, we have what is called an action potential if it reaches what we call threshold. And each neuron has a threshold of excitation. When that happens, when it's reached, when a neuron becomes depolarized enough and the inside of it becomes positively charged enough, it will trigger what we call an electrical event or an action potential. When this happens, a signal is sent in the axon down all the way and goes through the terminal buttons out into the synaptic space or that gap between neighboring neurons. Now let's look more closely at how that actually occurs. So the exchange of ions in the membrane. What tends to happen if we think again about the magnets and we think about positive and negative poles? Naturally, negatives and positives are more attracted to each other, and positives and negatives repel. So what tends to happen is at rest, this is a membrane of a neuron at rest, molecules tend to kind of evenly distribute themselves. So you have the positives and the negatives here that are more attracted to each other, and the negatives and the positives here that are more attracted to each other. So they tend to distribute themselves evenly. And if this happens, the cell will remain at rest or at resting potential. Now, what happens that changes this and eventually causes the inside of the cell to depolarize or become more positively charged is called electrostatic pressure. This basically is just a term to describe the attraction or repulsion based on charge. That is, again, negatives and positives are attracted, and then positives and positives, for example, tend to repel. Now let's take a look at how electrostatic pressure maintains resting potential of a neuron, or maintains that negative 70 millivolt measurement on the inside of the cell in the fluid. So first, let's take a look at A minus inside the cell in the intracellular fluid. A minus, again, that's the organic anions. These are unable to pass through the membrane of the axon because of their structure. So they can't actually leave the membrane. So these are going to hang out inside the membrane. Now then we have also potassium that's concentrated inside here. Now diffusion, which is just a force that tries to push outside, diffusion will try to push the positive potassium outside the cell. But electrostatic pressure actually pushes it back in because the outside of the cell is more positively charged than the inside. And because potassium is also positive, two positives are going to repel. So the positive concentration out here and the positive potassium in here are going to repel each other. So the electrostatic pressure is going to push it back inward. And this forces a balance in the fluid. And because A minus is a bit more concentrated, we have a negative concentration of negative 70 millivolts when the cell is at rest. Now, if we go further, we also have chloride, which is a negative ion out here on the outside. Now, the force of diffusion, again, that's that force that's going to try to push. It's going to try to push chloride inside the membrane. Now, if you remember, the inside of the membrane is negatively charged. Chloride is also negatively charged. So what's going to happen when we try to push negative chloride into the negatively charged intracellular fluid? Exactly. Because of this electrostatic pressure, two negatives are going to repel. So even though the force of diffusion is trying to force chloride in, because it's negative and because the inside of the cell is also negative, they're going to repel each other. And this again forces that balance of resting potential. Now lastly, we're going to look at sodium. Sodium is again concentrated on the outside. It's positive. Diffusion, again, that's that force that's going to try to push, tries to push it inside the membrane. Now remember inside is negatively charged, so electrostatic pressure won't prevent it from actually going inside the cell membrane because sodium is positive. Now we can ask the question of what stops sodium from continuously flooding into the cell. If the inside of the cell is negative and sodium is positive, and diffusion is pushing the sodium into the inside of the cell, what is stopping sodium from just continuously flooding in the cell? That's what we call the sodium-potassium pump. And you can take a look. Here is a cell membrane, and here is that pump embedded in the cell membrane. Basically what this is just protein molecules that form this pump. And what this pump serves to do is basically regulate the exchange of sodium flowing in and out of the cell. So basically there's a trade here. 3 sodiums out for every 2 potassiums that are pushed in. And this basically is going to happen kind of continuously to keep the sodium from just flooding in the cell continuously. This exchange of positive and negative is an attempt really to try to rebalance the fluid distribution and keep sodium from just free flowing into the cell. Now let's take a look at action potential. Now, as we were just talking about sodium, sodium is the one on the outside of the cell that by force of diffusion is being forced in, and electrostatic pressure will also allow in because of its positive charge. And again, remember the inside of the cell is negative. So sodium comes in to the cell through diffusion and again electrostatic pressure as well, because positive and negative are attracted to each other. So let's take a look at it in terms of the millivolts here. So remember, at rest, our neuron is going to be at negative 70 millivolts. And here we have drawn in the threshold of excitation. So once the charge is changed enough or is positive enough that it passes this threshold, an action potential will happen. So how does this occur? First, the sodium molecules or the sodium ions from the outside of the cell begin to enter the membrane of the cell, which will make the charge of the inside of the cell more positive. Once it passes threshold, we will have an action potential because again, the interior of the cell is becoming more positively charged. Okay, so sodium is coming in and then potassium starts to leave the cell. Because if you think about the inside of the cell, sodium is coming in, making it more positive. Well, now we need to rebalance. So potassium, which is positively charged, and on the inside of the cell will start to leave the cell. So as the inside of the cell is becoming more positive ions are being forced back out. And again, this is that sodium potassium pump. If we take a look at here closely, that is what is controlling this. And basically what happens here, because the cell becomes more positive with the entrance of sodium, and then to rebalance, potassium is forced out. So positive potassium is forced out to try to make the membrane on the inside of the fluid on the inside of the membrane more negative again. When this happens, this will return the charge of the inside of the cell back to being negative again, which is also called repolarization. So depolarization is when the cell is becoming more positively charged with the influx of sodium. Once that happens, potassium begins to be forced out, and the cell then becomes more negatively charged again, so it's depolarized, and then goes back to resting potential again. During this time, once action potential has happened, the sodium channel becomes blocked, so no more sodium can come into the cell. And also, then the potassium channels also close. So basically, the cell resets itself. Now let's take a look at the rate law for action potential. So action potential, again, is just when the inside of the cell becomes positively charged enough or more positively charged that it meets whatever threshold it has. And once that happens, an action potential occurs and the cell will send a signal down its axon down to its terminal buttons to be released to other neighboring neurons. Now, an action potential is an all or nothing response. It either happens or it doesn't. There is no range of intensity or strength of an action potential. It's either an on or an off switch. Either threshold is met and the action potential happens, or it doesn't. Now, what happens though when we have a weak or a strong stimulus? If action potentials aren't measured in terms of intensity, well, then how do they differ when we have a weak stimulus versus a strong one? Basically, what happens is that more action potentials occur. So this will happen through more rapid firing or rate of occurrence. So how many action potentials are happening? And we can take a look at that here. So if we have a weaker stimulus, we have fewer action potentials happening. But if the stimulus comes on and it's a strong one, we have way more action potentials happening. So action potentials are kind of measured more in terms of rate of occurrence. So how many of them are happening, not of any measurement of strength of them happening. Again, it's either on or off. It either happens or it doesn't. Now, I've mentioned synapse several times. Now we can take a closer look at what synapse actually looks like. And again, it's the gap between a neuron and its neighboring neurons. Or more specifically here, it is the gap between the terminal buttons here at the end of the axon of this neuron and the dendrites of the neurons around it. Now, during an action potential, the signal sent will come down the axon all the way down to the terminal buttons, and a signal will be released from the terminal buttons. It will be a chemical signal, and it will be picked up by the dendrites of the neighboring neurons. Now, there again is a small space between the terminal buttons and the dendrites. And if we take a closer look here, we can see that space. Here is a more detailed look at that. So here we're taking a look at the terminal button, and we're taking a look at the gap between that terminal button and the neighboring neuron here. So the signal will be traveling down the axon to the terminal button, and once it gets to the terminal button, it will leave in the form of a neurotransmitter. Now, what neurotransmitter that will be dependent upon what kind of neuron we're talking about, but the synaptic vesicles here will bind and will release the neurotransmitter into the synapse or the synaptic cleft, this space here between this terminal button and the neighboring neuron, and it will then be taken up by the receiving neuron. So that is basically how an action potential happens and how two neurons communicate with each other. Here we'll take an even closer look. So synaptic transmission is just when messages from one neuron are going across the synapse to a neighboring neuron, like what we just took a look at in the previous slide. Now, the presynaptic cell is just in reference to the sending cell. So which neuron is sending the message? That is the presynaptic cell. And the postsynaptic cell is going to be the receiving cell or the receiving neuron. Now, within the synapse, we'll take a look more closely here. So here we have the terminal button of the sending neuron, and here we have the reception site of the receiving neuron. Now, in the synapse or the synaptic cleft here, so that gap between the two, this converts the electrical signal being sent by the sending neuron into that chemical signal in the form of a neurotransmitter. The postsynaptic receptors will detect that presence of the neurotransmitter. So reception sites here on the receiving neuron will detect the presence of the neurotransmitter in the synapse. And after bonding happens, so after the neurotransmitter binds with the receptor sites here on the receiving neuron, the signal will be converted back into an electrical signal inside the receiving cell. Now that we kind of have an idea of how an action potential occurs, let's take a look at what happens when the receiving cell or the receiving neuron receives the signal from the sending neuron. We say that that receiving neuron has what is called a postsynaptic potential. What this means is that after an action potential has happened and this receiving cell has received a message from a neighboring cell, that will either result in an excitatory or an inhibitory potential of that receiving cell. And that is largely determined by what has been sent by the sending cell. So was it positive or negative? So the exchange of ions from the sending cell based off of the neurotransmitter that was sent into the synaptic cleft and was allowed to bind with the receiving cell, that will determine what ion channels are open in the receiving cell. Now, depending on what comes into the receiving cell, that will alter the charge of the receiving cell. And again, remember, at resting potential, the inside of the cell is always negative 70 millivolts. So if we have what is coming in the cell to be more positively charged ions, then this will be an excitatory postsynaptic potential, or ESPS. This will lead to that depolarization, or the inside of the cell is becoming more positively charged, so we're more likely going to have an action potential, an excitatory reaction by that cell. Alternatively, we could have ion channels that open that allow more negatively charged ions to try to come in through diffusion. And if this happens, the inside of the cell is going to become even more negatively charged. We call this hyperpolarization. So if the cell is becoming more negatively charged, then it's not going to meet threshold, and so that's going to be more of an inhibitory response. So the excitatory is going to lead the cell to be reactive and an action potential to occur, whereas the inhibitory or ISPS possibility will again make the inside of the cell more negative, threshold won't be met, no action potential. Now again, all of this is determined by what ion channels open in the receiving neurons when they received the signal from the sending cell. These postsynaptic potentials of the receiving cell, whether they're excitatory or inhibitory, are kept brief. And this makes sense. We don't want the charge of the receiving cell to continually be in flux and changing all of the time. And that's what will happen if the neurotransmitter that is sent by the sending neuron is allowed to just stay in the synapse. So what happens is that neurotransmitter is removed from the synapse by one of two processes, the first of which is called reuptake. So when this happens, the sending cell, the terminal buttons of the sending cell that release the neurotransmitter will then rapidly remove or reuptake that neurotransmitter back in. So the terminal buttons that sent the neurotransmitter will take it back in, away from the synaptic cleft. Now, another option is called enzymatic destruction. When this occurs, an enzyme is released that will destroy the neurotransmitter completely. So it's not taken back in by the sending cell, it's just destroyed. And this occurs when we have neurotransmitters that contain peptides. So for example, acetylcholine is a neurotransmitter, and we will discuss acetylcholine when we talk about neurotransmitters in more depth later in the semester. But this particular neurotransmitter contains peptides. And because of that, enzymatic destruction is what is needed to remove that neurotransmitter. So the postsynaptic membrane, which is again the membrane of the receiving cell, will actually contain what is called acetylcholinesterase. That is going to be the enzyme that breaks down acetylcholine, and it will break it down into its two parts, choline and acetate. So it completely breaks down the neurotransmitter, so it's no longer affecting the potential of the receiving cell. So what you need to know is that the postsynaptic potentials, or the potential of the receiving cell, whether it's excitatory or inhibitory based upon the ion charge, or the charge of the ions that's coming in the receiving cell, and how that changes the polarization or depolarization of the inside of that receiving cell. Regardless of that, these potentials are kept brief, and they're kept brief because the neurotransmitter that's released into the synapse doesn't hang out for very long. It doesn't hang out very long because of one of two processes, reuptake or enzymatic destruction or deactivation. Thus far in this lecture, we have just focused on action potentials. And again, action potentials are happening when cells are neighboring or are very close to each other. There are other types of neuron communication that happen when cells aren't as close to each other, so a more widespread communication. One form is chemical communication. This happens when with neuromodulators. So neuromodulators are basically chemicals that are released and dispersed farther and more widely throughout the body. This basically happens to modulate activity of neurons, specifically in the brain, most likely. And this may happen when general behavioral states are affected. And most of these that are released are peptides or amino acids. And again, these are meant to be secreted in larger amounts and for diffusion of longer distances. So they're really meant to affect things like general behavioral states such as vigilance or attentiveness, fearfulness, sensitivity to pain, for example. Other types are peptide hormones. So these are specific target cells that respond to the presence of a peptide hormone. So these affect the activity of these particular neurons. And the target cell that's going to receive these contains receptors for a specific hormone. So a good example of this is testosterone, for example. Testosterone is not transmitted through an action potential, for example. Also, steroid hormones are transmitted in a different way. So steroid hormones really initiate change of a physiological process, as you can imagine. They also easily pass through a cell membrane, so directly go into a cell membrane. A good example is sex hormones. Now that we've talked a lot about neurons in both this lecture and the first part of the lecture for chapter 2, we've discussed form of neurons. We've also talked about their function, and we've really focused in this lecture on communication. We've talked about action potential as well as in the previous slide on some ways that chemical communication happens as well with neurons. In this last bit, I wanted to focus on diseases that affect neurons. So first we'll take a look at ALS or amyotrophic lateral sclerosis. This affects what we call both upper and lower motor neurons. Now typically, upper motor neurons are more responsible for motor movement, whereas the lower motor neurons are more helping you to not move too much, basically prevent excessive movement. So if you have a disorder of both, you're kind of going to get a mixture of both symptoms happening here. Primary lateral sclerosis, this is a disease of the upper motor neurons. So what you might have here is spasticity or kind of tremors, things like that. Whereas something like progressive muscular atrophy, this is going to affect the lower motor neurons in the spinal cord. And what you might have here is a lot of muscle atrophy. And then progressive bulbar palsy, this is going to affect the lowest motor neurons in the brainstem. These are going to be the ones that are most affected. And again, this will tend to cause more atrophy. So patients with this may present with slurred speech, chewing issues, swallowing problems. As we move forward in the semester, we will talk about brain areas in more depth, and we will focus on the brainstem. But just know for now that the brainstem is the oldest portion of the brain, and most basic life functions are really associated more with the brainstem area. This would include sleeping, breathing, swallowing, chewing. So it makes sense that this particular disease may present with issues of speech, chewing problems, swallowing problems. Also mildly abnormal signs in the arms and the legs as well. All right, folks, that's all for now. Thank you for hanging with me through this lecture. I know that this material can be a bit dense and a bit sticky to understand. I hope that this lecture helped clarify some things a bit for you. I'll also include some supplementary videos for you to take a look at that will also hopefully help you further understand just basically how neurons can communicate with one another, really focused in on action potential in particular. I hope you have a wonderful week, and I will be back with you soon. Bye-bye.